Go ahead and get started. We see a few more of the attendees have logged on, and I'm
Oh,
sure
he's
more
here.
will.
He's here.
Here he is. Perfect.
Yeah, perfect timing. Yeah.
All right, Berkeley, we were just getting started. Just welcoming everyone who's joining us here live, and then welcoming those of you who are going to be viewing this online, either through the registration link or on our website. It's a pleasure to welcome you. So my name is David Buckner. I'm an associate professor here in the Department of Genetics and Genome Sciences at Case Western Reserve University. And I'm also the program director for our brand new master's program in computational genomic medicine. So we've put on a few webinars already. So if anyone hasn't seen them, I would encourage you to go to our website, and you can take a look at them there that have sort of gone through the master's program in more detail in terms of what classes you'd be taking, what research opportunities there are, where you can do research opportunities, things like that, what sort of background we're looking for, what kind of jobs are available to folks after they graduate the master's program. The last one, we also had Dr. Tom Laframboise, who's directing the computational genomics course. He kind of went through the syllabus in detail, so that was a really good one to take a look at to see what one of the classes you'd be taking, what they'd be learning, and different things like that. But a lot of the questions we've been getting from a lot of different students are really focusing on kind of what the research opportunities are as part of the program. And I think this is really one of the strengths that separates us from a lot of the other programs that are out there, is really the opportunity to get in the lab and get some hands-on experience doing some cutting edge research. And so today we're joined by two of the other faculty in our department, Dr. Yan Li and Dr. Berkeley Greider, as sort of two representative outstanding faculty that we have in our department, that you'd be able, with different research focuses that they each do in their lab. But these are two different labs that you would be have an opportunity to work in, as part of our program. So we kind of invited them to join us for the webinar to talk a little bit about the research they do in their lab, what they're looking for in students, different opportunities, things like that. And so we'll welcome them here, and we can start if Dr. Lee, if you wanted to sort of introduce yourself briefly.
Hi, everyone. I'm Yan Li. I'm at the Department of Genetics, and my lab is interested in using different genomics approaches, such as function genomics, single-cell genomics, epigenetics, and integrate those multi-omics tools to study islet biology. So you can imagine islet is the insulin producing a mini organ. So we are focusing on the diabetes research. Hope we can have a very nice communications. And if you have any questions, let us know. Yeah.
All right, thank you. And Dr. Greider, do you want to briefly introduce yourself too?
Sure. My name is Berkeley, and I run a lab that is focused on doing genomics and epigenomics, not just for the sake of discovering new genes or new vulnerabilities for disease, but also really to understand drug mechanism of action. So we do a lot of drug development, and basic question is, okay, you give a drug to a cell, what genes are changing? How is the chromosome folding differently? Like, what's the actual way that the cell is taking that drug and responding to it in real time? And so we're opening up this sort of black box. So, previously you'd be like, oh, you give this inhibitor and then you get apoptosis or you get differentiation as like the output. But there's like this giant black box in the middle. Well, how in the world did that occur? And so our lab is focused on answering the why, like the circuitry of how those cell-fate decisions are made in response to therapy, which really is critical to understanding everything from resistance mechanisms to why certain cell types are sensitive to some drugs and not others, things like that. And most of it's done in my lab in the context of childhood cancer.
All right, well, great. Thanks very much, both of you, for joining us, and looking forward to hopefully a good discussion. I wanted to sort of start really in the beginning, right? So our master's program is in computational genomic medicine, and I sort of wanted to ask you, I think that means a lot of different things to different people. And so I wanted to start by asking you both sort of what does that mean to you to be in the field of computational genomic medicine? So Yan, if you want to start.
Yeah, I think my understanding is with the advance of these technologies. So no matter from the computational wise or is from the sequencing platform, so we are facing a lot of data. If you're thinking about, the genetics background in human, we are also digital based, right? DNA is the driver of all the protein, all the functions. So how can we integrate those informations, integrate with those new technologies for the future medicine, for the future treatment? I think this is this master program. This is our goal. We need to learn those new technologies and new tools and how to solve those problems in a big data level, and then prepare our students for the future career, including academia, in the healthcare, and also in the industry. Yeah, that's my understanding. Yeah.
Yeah, that's great. Do you have anything to add to that, Berkeley?
Yeah, I mean, computational genomic medicine is certainly a mouthful. I would say, we're entering a new era where computation is giving us a lot in the field of medicine that was previously not really tractable. So, some of the biggest tools that have come online recently are really just the beginning, such as AlphaFold, which taught us the three-dimensional structure of all proteins in the human genome based on first principles and based on machine learning prediction, which actually turned out to work very well. Well, there's a ton of additional problems that advances in computation are allowing us to begin to approach, such as what about when you have not just one protein, but let's say 4 or 5 proteins? What happens when those proteins are interacting with DNA or RNA? Like, how does that change their three-dimensional structure? Does that create new binding pockets where we might imagine fashioning a drug to go after that interface, which you can't detect using things like crystallography for various technical reasons. But with computation, you might actually be able to begin building robust models and finding places to actually use the power of medicinal chemistry fueled with computation as a sort of like widening aperture of discovery to actually go after things which before we could never imagine going after. That's just one example. There are many. So, the amount of data that's out there, genetic information about which genes are essential in which cancer contexts or which disease contexts because of CRISPR editing technology, has given just an absolute flood of very meaningful biological information that has, for the most part, yet to be translated into medicines. Because we're living in an age where we're deluged with a tsunami of data of all different kinds. And one of the goals of my lab and the goal of this department in this program is to create the next generation of surfers for this tsunami of data, who can surf and fish at the same time, as it were, in this massive amount of new information, using the best tools. And the tools are getting better all the time. I mean, maybe, I don't know if there's a spot later we can talk about this, David, but I think harnessing agentic AI tools for computational genomics and medicine is going to change the way we do research. It is already changing the way that we do research. And that's something I certainly am exposing my students to. And I think it's a really fun time to be interested in this field.
Yeah, it was great. I was actually going to ask about that later. Let's do it now, since you've kind of brought it up already in your answer to that question. How do the students in your lab use AI? Do you see this as a threat to computational genomics? Is it going to replace, sort of what we're what the students are doing, or is this going to augment sort of what we're training? How do you see AI? How do your students use it?
Yeah, so it's like this. Having a personal computer is kind of like having a bicycle, right? You have your brain in your hands and your bicycle. Now you could walk from here to Tennessee, or you could get on a bicycle. You're going to get there a lot faster on a bicycle. That's what a computer is. Well, AI and the tools that are available now is it's kind of like hopping in, a Ferrari that can go 100 miles an hour to get you there a lot faster. So you still have to know where you want to go. You have to know the questions you want to ask. You have to be aware of the data that's available. You have to have the biological insight and like have an aim. That's something that the students come up with, and the students, are constantly evolving and updating their questions as they're pursuing them. But I look at it as just a vehicle to accelerate your ability to write code, to analyze data, to debug. The the fact of the matter is, like, we've got all of this code already written in videos to like explain how do you run an RNA sequencing pipeline. Okay, simple, right? But the downstream analysis of that is like 40 different scripts that other people have written. You're going to go and learn each of those. Or now you can actually just go with, as long as you're aware of like, what are my samples? What's my question? What are the kinds of things I want to do with my data? You can just explain to these things like Opus from Claude. You can explain, here's what I'm looking for. Here are some of the ideas that I have, and here's the structure of the data. Now I want you to write an R script wrapped in a shell script that produces PDFs of, for these three figures that I know I want. And why don't you, by the way, come up with 2 additional figure types that I haven't thought of yet that look at the data in a different way. And it'll write that code for you in about 2 minutes. You run it, see the output, but it's basically just squeezing the time gap between idea and seeing the results of the output of your analysis. So I view it as a very important accelerator, because it still requires intellectual guidance. It's not going to do your PhD or your master's or your research for you, right? You have to be the driver. And so it just allows you to just drive faster.
Yeah.
That's how I view it. That's how we use it.
Yeah, your thoughts?
Yeah, I agree. I think the AI definitely can boost our productivity. However, I think the major difference from us is the question. The question is always, we need to ask the question, what's the most significant goal we want to reach, and then we can use the tool like AI to help us. And I think another layer is the decision. So at the end, hopefully, still we are human. We human make this decision and makes this judgmental direction. You're gonna evaluate, or we're gonna evaluate, whether the AI's the analysis or the productivity of the result it's reasonable, it's correct, right? At the end, I think we are making this call because it's the machine is not always correct. We know it's sometimes it's a lot of nonsense, but I agree it's a very can boost up our productivity, and we need to learn how to use it. It's a very powerful tool. Yes.
Yeah, actually, I couldn't agree more with both of you. It's been a great tool, and it's just speeding up what we can do, not really replacing.
Yeah.
Not replacing it. So I wanted to ask you both, can you share an example, like a specific example of a recent project that a student had in your lab, sort of what kind of data sets they were looking at, sort of what unmet clinical need they were trying to address, or specific advance on that? Jan, do you want to start?
Yeah, so there are several projects or directions in my lab. The first one is we use a pluripotency stem cell to differentiate them to these islet-like cells that can produce insulin, because we know this platform, this method is we believe hold a great promise for type 1 diabetes treatment, because in those patients, those beta cells in the islet are specifically destroyed. That's why the T1D patient, they cannot produce insulin, and then they need the insulin injections. So we're hoping by using this unlimited resource from this derived islet-like cells, so we can transplant those cells to type 1 patients. However, the gap is right now the technology is still have a big gap. For example, we know this in vitro differentiated cells is not exactly the same as our primary human islet cells. So what we do is in the lab, one thing is we use the genomics approach to map this molecular to characterize the differentiation pathways. So basically, in every step, we map the epigenetic features and the expression features, and then to identify what's the best driver. We could drive the cells to the correct lineage, to the function islet cells. And the other direction along this line is we are hoping to integrate genetics and epigenetics information, see whether we can predict the differentiation efficiency, right? If you think about it, if in the future we want to do the cell therapy, but without knowing whether the cells can be efficiently differentiated to the organ that we want, then we are hoping, at the very beginning, we could know, what's the successful rate. So we are hoping to use this computational approach, integrate the information from the genetics and epigenetics, and then at the very beginning, see whether we can predict this differentiation efficiency. And then another line of the research is we know there's a disease difference, variations, or between healthy and the disease patients, right? For example, in our cases, we focusing on, identify the difference between type 1, type 2, or obesity patient within their islets. So what's the difference compared to the healthy persons? So in knowing of that, can help us to understand the mechanism and then identify the better drug targets. So in this directions, we collect the human islets from the deceased donor, either from healthy donors or from diabetes donors. So we performed like single cell RNA-seq, single cell ATAC-seq, or even the high-seq, like Berkeley just mentioned, to study the chromatin organizations. But all of this is, like I said, is trying to identify the difference, the disease, see whether we can identify the disease-causing genes at a single cell level, and the genetic risk factors to help us better predict disease risk. For example, like a T1D right now is still, we do not have a very early, we do not have a very robust approach to detect, those early events for the disease. So those are we are relying on this genomics approach. And then of course, we have to integrate both computational part and wet bench technologies, and then to push these things forward. Yeah, and then the third part of the lab is we, in collaboration with Dr. Fulai-Jin's lab, we also trying to develop a genomics approach called single cell Hi-C. So we are hoping to study the chromatin structure at the single cell level, and then also integrate this genetic analysis with this chromatin structure information. Yeah, that's pretty much what we are interested, right now working on. Yeah,
that was great. Thanks. And then Berkeley, same question. A specific example of a project that you and your lab have been working on that has some clinical implications?
Yeah, well, there's several. I think I'll just give one to make it brief. So, we do childhood cancers, but there's many, many, many kinds. And we focus on the solid tumors, often sarcomas, as opposed to leukemias, which occur in the blood. And, a lot of times the exact same disease, or like it'll have the same name, like rhabdomyosarcoma. Well, there's like 4 different kinds of that. And clinically, some of them look identical, but genetically they're driven by totally different things. So you might think, well, that probably means that you should give a different drug based on the genetic problem that's actually driving that cancer. So our lab defined, for example, that you should give MEK inhibitors, which is basically an inhibitor of a signaling pathway that's hyperactive in some subtypes of this disease. That particular therapy works extremely well and selectively for that subtype, genetically defined subtype, which is actually translated into clinical trials for that one. There's another subtype which has a mutation in a totally different pathway, and we've gathered all of the genetic and mechanistic data necessary to convince the National Cancer Institute to start a clinical trial for that targeted therapy with this other, it's called mutant MyoD rhabdomyosarcoma. Very, very rare, but extremely deadly disease. And then for the same disease driven by this fusion Frankenstein transcription factor that's got the head of one transcription factor and the legs of a totally different transcription factor, called PAX3-FOXO. Well, that driver for yet another subtype of the same disease, that's a genetic event, and it turns out that cells with that particular genetic abnormality are extremely sensitive to a handful of targeted agents that have no effect on the other subtypes. So we did a lot of drug screening to kind of figure this out, but also a lot of genetic screening where we'll knock out all the genes in the genome, or maybe we'll just pick like, oh, I just want to knock out all of the epigenetic genes and then see across a panel of like 12 cell lines, what genes when knocked out only killed this subtype and not the others. So you kind of put these things together and you get a picture of what each subtype is very reliant upon in a kind of a selected way. Now you want that because you want to, gear a drug towards the clinic that's going to be, let's say, 10 or 100 times more potent at killing your specific type or subtype of cancer, such that you need 10 times less drug or 100 times less drug. That way you can actually give an amount that doesn't start to cause side effects to a patient. You want this sort of therapeutic window. And genetically defining this relationship is what we've done for many subtypes, but it requires a lot of computational power to clearly mark all of the to put all the signs up on the road properly, right? To know exactly what's what. And we've done that, and we're continuing to do that with new cancers. And this has already made it into the clinic for a couple of diseases, and we're trying hard now for some new drugs that we're developing. And we're able to basically say these are the genetic biomarkers of response. So when you design your clinical trial, this is a genetic test that you must do before saying this patient should get this drug. And that's, I'm not a medical doctor, but, I collaborate with upwards probably 30 different MDs at different institutions to sort of feed them the information that they need to design trials properly. Does that make sense?
Yeah, it was great. So there's a really great, actually a great description of a couple of the different research projects that would be just, two or three of the many projects that would be available to students in this master's program here. Here, okay, so you talked a little bit about the science, the big picture about the project. Like when you get into it, what does the day in the life of a student look like in your lab as they're working on these projects? What do they do all day?
Well, you want me to start?
Yeah, sure. Go ahead.
All right. So it depends. We have some wet, some dry, and some moist lab members. Um, so the wet lab biologists are, they're pipetting, they're growing cells, they're plating them in wells, and doing different kind of genetic or drug perturbations, and then doing readouts of different kinds. Computational biologists in my lab, they spend a lot of time analyzing data. So they're just, they're talking with me on a whiteboard like the one behind me. We're drawing out ideas. We're saying, okay, I bet you if you looked at the data in this way, you'd see this kind of a trend, and here's the plot that you would want to make in order to get that, in order to test to see if that's real or just in our imagination. And then they'll go and they'll say, okay, this is all the data that I have, and they'll sort of pull out their list of sample files that are relevant to the question. Then they'll write up some scripts that reach into our sample directories and pull out different kinds of data associated with each of those samples, and then merge them together. And they'll create these big data frames, and maybe they'll then take gene sets that mean different things, like a set of genes associated with muscle differentiation for a muscle cancer. And they'll say, oh, I'm going to take all these samples and now I'm going to, do some subsetting to look at trends in this particular set of genes for all of these samples and see like, which of the tumors are most differentiated or least differentiated, something like that. But a lot of it is, looking for trends and thinking about how to interpret or how to do a lot of it's dimensionality reduction, because we're always dealing with large cohorts of hundreds of samples and 30,000 transcripts, and so you've got to be able to like massage the data, compress, summarize, extract. So there's just a lot of analytical steps that most of my students will spend time either coding directly in R or RStudio to sort of work on the visualization part of it. In my lab in particular, we automate as much as possible. So the vast majority of the heavy lifting bioinformatics that's kind of rote and routine, students don't need to learn that because that's all done, and all they need to do is learn how to run it. They just push a button and it does 8 hours worth of computation on their part, and then they get to actually dive into the specifics. So a lot of it's a conversation with the data, how I like to put it. They ask a question of the data, they make 10 different kinds of plots to look at different trends, look to see, oh, is this gene that I think is important, is it going up when I, make this contrast between this set of patients and that set of patients? And if it is going up, then maybe they'll say, oh, what other genes are correlated with it? Whatever. And if their hypothesis is wrong, then they'll go back to square one. And then maybe I'll say, hey, you should just go spend an afternoon reading 3 or 4 papers that are on this topic to expand your ability to come up with new ideas or to ask new questions of the same data. So that's also, I think, a really important part of the development of people in my lab is actually critically reading and learning, which is often a lost art. So we try to encourage that.
I want to ask you both too. What do you look for in a strong application or in an interview for this program specifically?
Jan, you want to take the lead on this one?
Oh, okay, sure. So, well, I think the most important thing is the motivation. That's the first. If you are passionate about the things that you are doing, then I think you have a higher chance to do it well. So I think, and also, you're gonna enjoy it. So I would say the motivation is the most important thing. And of course, if you have some relevant background, but not necessary, it has to be, computational part. Because nowadays, for our program, we've already started to teach and practice assemble this class for 2 years, I think. Yeah, I think how the class designed is really, help students to learn from the, baby steps. So it really helps. So if you have some relevant background, for example, biology background or some computational background, or even some remote related pharmacology background, they all helps. And here for this program, I think how the class designed can help you to, from the beginning and then transit to, a project driven, and then eventually, be independent. So I think motivation, interest, very important. And if you have some relevant background, it would be a plus. Yeah.
Yeah, Berkeley.
Yeah, I mean, I think the most important thing is that you really want to launch yourself forward into this space. Qualifications will only get you so far. Experience will only get you so far. I think that intrinsic motivation and like a vision, a passion, like a excitement about what's happening in the very near future and in the future, like really wanting to put yourself in a position where you can ride this wave that's rising, in these fields. Like you've got to have a desire for that. I think that's what separates the students who are going to come in and just kind of check the box versus the ones who are going to come in and make a difference. And so that's what I live.
Yeah, I'll just echo that what both of you said too. Is our program doesn't really have any specific prerequisites where you don't have to come in with both computational or biology experience. It was really kind of designed to, if you come in with some biology experience, we can teach you the computational part. If you come in with computational experience, we can teach you the sort of genetics and genomics part. And then, of course, as part of the application, every applicant has a chance to write a statement to kind of describe, their motivation, what they're interested in the field in, and then interviews as well can kind of get those across too. So those are good chances to express what both Jan and Berkeley kind of nicely mentioned, to mention that sort of motivation part and really wanting to jump into the field. Another question. So I guess it's sort of what's special about CASE, right? What makes it a great place to come learn to be computational, get involved in computational genomic medicine here.
Well,
Yeah,
I
do you want to
think
start?
yeah, I think we have a long history for our department, the genetics department, we have a long history of doing the research related to the computational work. And we also have a long history of training the students or postdocs for that expertise. I remember, I think the student from our department got the HHMI fellowship, right? Doing the epigenetics and the genomic study on cancer study. And my first student, Chen, he came with a biology background without a computational expertise. And then after graduate, he went to MIT as a postdoc, and now he got a faculty offer from University Washington. So I think those are very good examples showing, I think in this area, we integrate both the biology and computational and the genomics training, very well. And this is our strength. And we are very proud of our students and the trainees. And I can tell you they are very happy here too. Yeah. So, so if you are interested in this program, you can also, I would encourage you to talk to our students, our postdocs, all the people here, and also I think the whole environment is also not only about research. We have a music institute, the art institute. It's a very vibrant and it's a fun place to be a student. Yeah.
Yeah, I'm sold. That was great. Berkeley, do you have anything you wanted to add to that?
I agree with everything that Jan said. I think I would just emphasize the fact that if you're coming in and you want to do computational genomics, in some sense, you could do that in your underwear on your couch, right? Without anyone telling you what to do. But if you're here, what you get access to is mountains of unpublished information, unpublished data. You get access to working directly with people who will generate new, actual, real data for you in response to an insight that you generated from your computational analysis. You can't do that sitting on the couch. And you can't do that at at many other institutions. So we have the ability to create a very vibrant, kind of real life, what's the right word? This is a fast acting, fast moving, intellectually intense, highly collaborative, forward looking group of well funded and very curious and highly skilled people who are all around you. And that's something that's why I'm here, because we are able to do infinitely more in the environment that is here in Cleveland that I get really excited about. I get so many of my ideas, or so many of the things that I'm working on now, I never would have thought of if I didn't have a coffee with XYZ individual, or there are instances where I've seen something in my data and I did not know what it meant, but I was able to, send a cold email to someone I've never spoken to who's an expert in this field that's relevant, or who's an expert in this gene that popped out of my analysis. And then we start a collaboration and, he starts doing experiments and it opens up whole new avenues of possibilities. And those are the kind of things that make Case Western special, and meaningful and translational.
Yeah, that was great. So we have a little bit of time left. I wanted to sort of switch gears. We had a number of questions that students sent in, students that registered, that are here online now. So I just wanted to give you a chance to answer some of the questions that they sent in. So the first question we got was, how do students in the program build enough computational depth if they come from a more biology-focused background? Actually, before either of you answer that, I'll sort of answer it a little bit. I would recommend taking a look at our last webinar. It's up on our website right now. And Dr. Tom LaFramboise really goes through in detail the syllabus and of the Intro to Computational Genomics class that every student will take in their first semester here. He talks about how there's really no expectations to come in with a computational background, and goes through like literally day by day sort of what you'll be learning and how you'll be developing those computational skills. But I think we've all sort of had students kind of in our lab that started working on computational projects and came in with no computational background, and they get it from the coursework. They get it from interacting with students around them in the labs, that come with the experience, and from directly interacting with the faculty here. So I guess under Berkeley, did either of you wanted to add to that too?
Yeah, I agree. I mean, I think the setting of the class is really helpful. It's just like I said, I have many students came with a biology background, and then after taking those classes, and also by doing practice in the project, so they equipped with this the skills to do the data analysis, and also I think for me, when training the students in the lab, usually I gave at least 2 projects to one student to make sure they have enough exposure to different skills, and also to make sure they have a good learning process. At the end, one of the projects, or hopefully both of them, could work out, and then to smoothly do the graduation and then move to the next step. So I think all of this will help the students who came with a biology background, and then during this several years of training, you could also master the computational skills, and then to make sure your future success and your future career development. Yeah.
Yeah. It's easier to teach, in my experience, it's easier to teach computation to a biologist than it is to teach biology to a computer scientist. So I would actually say that's an advantage, especially because actually practically accomplishing computer science tasks is less about knowing how to code and more about understanding data structure, understanding what is a read, what is read depth, what does it mean? Like, what does genomic alignment mean? What does variant allele frequency mean? Like, you have to understand those concepts, but like the actual, barrier to entry, which used to just be being able to write syntactically, error-proof code that will do what you want it to do. That used to be a huge bottleneck that would take students years to, get operational on. And now that barrier is largely reduced. And so I think, we'll give you all the skills that you need, and it's something that, you become a lifelong learner. I never had a computer science class in my life. I never had a genomics class in my life. As a matter of fact, I'm a chemist, and as a master's student, you're coming in as someone who has a biology background and you don't have any computation. Well, as long as you're okay with math, like if you're not afraid of math, and you have a biology instinct where you know how to ask interesting biological questions, then you're going to be fine. Like all the rest of the stuff kind of takes care of itself. Like if you have a good question, there are people all around you who will give you every tool that you need to get to your answer. And it won't actually be as difficult as it looks from the outside, I'll say.
Yeah. Next question we got was, what kind of careers do graduates most commonly go into after finishing the program, especially if they do not want to do a PhD immediately? Yeah, so that's one thing students can do is to go on to a PhD program, MD program, other sort of postgraduate programs. But what else have students in your lab gone on to do?
Well, I know people can develop the business sense, right? Go to the industry, and I know people went to consultant company, right? Because they also need this biology and the genomics expertise. And the government, right? For example, FDA, NIH, they also need the people with those expertise. Of course, healthcare, for example, insurance company or hospital, or it's even some company they provide the service to the hospital. They also need those expertise because people need to they need the people to understand those data and who has this expertise. So and of course, if you think about the industry like a pharmacology, a pharmacist, and all the health related, even food company, those industry probably also relevant to this. Yeah.
Berkeley?
Most of the students in my lab have gone on to do medical school, so
That's great.
So I think it's very different. I have a young lab, so not too many people have left. I've sent out a couple of people, mostly into academia, but I will say that there's a huge appetite for master's level computational biologists to just come and do research in both academia and in industry. I think especially if you've got the right set of skills and the right mentality, I think there's a lot of opportunity in both. You don't have to get a PhD to do meaningful research or just meaningful work in a whole variety of contexts. And so, for example, I just hired a master's student to join my lab and do nothing but computer science. She doesn't need a PhD. She just is excited to come, crunch some numbers and ask some questions. So that's all I have to say about that. But again, my lab's only 4 years old, so hopefully in a few years I'll have a more complete answer as I graduate more and more people.
Yeah, and you kind of touched on this next question a little bit. Someone asked about potential applications of this master's degree program within the business aspects of medicine. I think it was something you kind of touched on a little bit with some different sort of career opportunities. Yeah, I think, in terms of the coursework, we don't directly have any business classes or anything like that as part of the master's program. But obviously, if you're going on to do an MBA or something like that, sort of something that could set you apart, in that aspect is sort of having the specialized knowledge.
Exactly. Yeah.
Having the science to sort of back up, what you would go on to do your MBA, or if you want to go into like pre-law or something like that, law.
Yeah, exactly.
Especially like something like patent law or something.
Yeah, exactly.
To have the sort of specialized scientific knowledge. Behind the business degree, behind the law degree.
Exactly.
Something that our program would address.
Yeah, because even when they make the business decisions, they need the people who is the expert to understand what's going on. So yeah, actually, I know my peers. I do have some friends. They want to, like you said, like patent law and as a consultant, and then it's pretty broad. Yeah.
Yeah, consulting you mentioned too would be a
Yeah.
big. Next question we got was, how much of the program is focused on hands-on analysis of real genomic and clinical data sets versus lecture-based coursework? I can take that one. It's sort of a, again, I would, I keep plugging the last webinar. I would recommend going to the last webinar where we really went through sort of all the coursework and the kind of whole, what you'd be doing in this program, which can last somewhere between a year and a half to, depending if you want to do it part-time, 3 or 4 years. But more often, I would say a year and a half to 2 years. It's really sort of split 50-50, where the first year is mostly focused on coursework, but beginning to do rotations, beginning to get in the lab, things like that. And then the second year is really focused on research, where you're almost in the lab kind of full time. So out of the sort of 2-year-ish program, it's about a year of coursework and about a year of research. So it's kind of split 50-50. That said, some of the classes that we offer are working with sort of real world hands-on data. Depending on how you count that could change the ratio a little bit too.
Yeah, I mean, like you say, the question said lecture based, and for the course that I teach, there's very little lecturing. It's like break out your laptop. Here's, go here, download these file types, and here are the scripts that you need to run. Here's how you run them. All right, now let's pause. Everybody do that first step. They do it, and then like 4 people have no idea why it's not working. We'll walk around and say, oh no, you see, this is you did this wrong in the command line. Here's the fix, right? Very very much real data, like real clinical or biological data that we'll download and analyze. And then once you get into a lab, if you do research in a lab, like it's all if you're in my lab, you're dealing with data that came directly out of tumors from kids with cancer. That's what gets plopped in your lap, and then it's your responsibility to teach us all something new that's been there that we didn't see. So
yeah, that's all the research projects, right? It's all generating new knowledge, right? We don't have any research projects where you're sort of sitting in a lab with someone's data set that they've already analyzed before, and you're just sort of rerunning the analysis to make sure that you can learn how to run a script or something like that, right? All of the research we have are going to be generating new knowledge and tackling new biological problems, and so yeah. Last question. Actually, last question was sort of, can I transfer into the program? I think rather than giving sort of general answer to that, I think if someone's interested in transferring into the program, they should just contact me directly, because that's going to be a sort of individualized answer depending on background, where you are, kind of what exactly, sort of how you're looking to transfer into the program. So my contact information is on the website, or my email is dab22 @case.edu. So if you're interested in transferring into the program, just shoot me an email and kind of introduce yourself and sort of tell me what your situation is, and we can tackle that on a case-by-case basis. So I think with that sort of concludes all of the questions that we have. Were there any sort of parting thoughts, Jan or Berkeley?
Well, I guess we are very excited as a faculty. I'm very excited, for this master program, and then I hope you can come visit us and even join us and then grow with us together. Yeah.
Yeah, thanks, Jan. And then Berkeley?
if you haven't been to Cleveland, you're gonna love it. I've lived in a lot of places.
Yes.
This is top of the list for a lot of reasons. Cleveland's great, and the people here are great. So there's a lot of non-science based reasons why I think it's a fantastic place to be.
Yeah.
That was great. Well,
cool. Yeah.
Thanks everyone for attending the webinar, for sticking through it to the end. And then, of course, a huge thank you to Dr. Jan Lee and Dr. Berkeley Greider for sharing a little bit about what their lab works on and their thoughts about computational genomics and Cleveland and all sorts of other topics. And again, if anyone has any questions about our brand new program, feel free to reach out to me. I know Jan mentioned before, kind of one of the great things can be talking to some of the faculty or even talking to the students. And so we can help kind of put you in touch with sort of current students in the department too, if you have any specific questions about that. And then again, my email is vab22, and a lot more information is on the website. And thanks very much for tuning in. All right.
Yeah. Thank you, David. Yeah.
Yeah.
All right. Take care, everyone.
Bye.
Thanks very much.
Yeah. Bye.